I'm Conor Houghton, and this is the second half of the first two lectures I give as part of the course PHPH20007 on computational neuroscience. In the first half, I talked about the equations for a leaky bucket. So there we saw that tau times the rate of change of h is equal to I, the input, divided by g minus h, where h is the height of the water. And the reason we studied that equation was that it turns out to be similar to the Leaky integrator and fire model we use as a simple model of spiking neurons. And when we were discussing that equation, we talked a little bit about how to solve it. We saw the solution for constant input, and we also learned that h, the height, performs a filter on the input. And so the input might have some temporal behavior, some temporal structure, and that gets smoothed out, a sort of local temporal averaging. That, I think, provides an insight into what it is that neurons do with their input. So this is a neuron, and it might sound obvious, but one of the important things about a neuron is that it has an inside and an outside. It's surrounded by a membrane, a bilipid layer. It has a salty fluid inside and a salty fluid outside. In fact, the fluids are salty in different ways. On the inside of the neuron, there's an excess of potassium ions. On the outside of the neuron, there's an excess of sodium ions. The K there standing for potassium, the Na standing for sodium. There are other chemical gradients, other disparities in the number of ions of one type or another, calcium, for example, and chlorine. But in this version of the story, we're just going to concentrate on sodium and potassium, mostly on potassium. Of course, because there are charged particles and there's a different number of them inside and outside, there's also a voltage difference. And so the inside of the neuron is at a different voltage, in fact, a negative voltage, around minus 70 millivolts, compared to the outside. And the story that we're going to tell, the story of the integrator and fire neuron, is the story of the dynamics of that voltage. In fact, this disparity is maintained by ion pumps. This is a picture of a cartoon of one of these ion pumps. They rock backwards and forwards, and as they, in one configuration, they take in 3 sodium ions marked with the yellow dots, and release 2 potassium ions. And then in the other configuration, they release 3 sodium ions and take in 2 potassium ions. And so the top in this diagram represents the outside of the neuron, where the sodium ions are being pumped, and the bottom represents the inside of the neuron, where the potassium ions are being pumped. And obviously, moving the ions doesn't come for free. These ion pumps absorb the energy carrying molecule in the body, ATP, and use that energy to create these disparities in the number of ions. Because, of course, by creating chemical gradients, which we'll talk about again, and potential gradients, that is differences in voltage, the energy is being stored across the membrane. And so it has to come from somewhere, and it comes from ATP. So afterwards, you end up with, as I said before, more potassiums inside than outside, more sodiums outside than inside. Of course, it's not that there's just potassium inside or just sodium outside. The ion pumps don't remove all the sodium inside the cell, but there is more sodiums outside than there is inside. It's important you remember which way around that goes, where the sodium is and where the potassium is. The way I remember it is, if you see here in the map, in yellow represents the United Kingdom. That has a K, like the K in potassium, and that's kind of inside for us, living as we do in England. And then around at least most of the United Kingdom, there is the C. The C is salty, and we always remember that salt most commonly is made up of sodium and chlorine. And so sodium on the outside and potassium on the inside. As we just discussed, there's as we saw in the ion pump, for every 2 potassiums that are pumped in, 3 sodiums are pumped out. So there's more. The disparity is greater for sodium than for potassium. Obviously, this isn't the whole story of transport of ions across the membrane, but generally speaking, there's more. The degree to which there's more sodium outside and inside is greater by a factor of 3/2-ish than the degree to which there's more potassium inside than outside. And because these ions are positively charged ions, that means that the voltage inside is negative. I mentioned before that we have this kind of typical figure in mind of minus 70 millivolts. Now, as far as ions crossing the membrane is concerned, that's not the whole story. There are other ion channels through the membrane. There are actually quite a variety of ion channels. There are active channels that we're going to talk about later that change their conductivity, the degree to which, or the ease with which ions can pass through them, according to the voltage or according to other events like binding. The voltage gated channels, obviously, it's according to voltage, but there are also channels that might bind to neuromodulators or neurotransmitters. Or even be sensitive to the concentration of calcium on one side of the membrane and the other. So there are ion channels that allow ions to pass through the membrane. Passive ones that we're going to talk about now, which don't change their behavior according to the local conditions. And then the active ones, whose conductivity might change according to, for example, changes in voltage or changes in concentration of neurotransmitter, neuromodulator, calcium, etc. And so here, obviously, this is supposed to show a potassium channel, and it's allowing potassium to flow from the inside of the cell to the outside. These channels are, to a remarkable degree, at least most of them, ion selective. So a potassium channel will only allow potassium ions in it by a factor of 1 to 10,000 and won't allow sodium ions to travel through. So that's the neuron. That's an inside and outside. It has more potassiums inside than outside, more sodiums outside than inside, and it has some conductivity, some permeability. And what we're going to try and do is write down an equation for the change in the voltage on the inside of the cell. This equation, the Leaky integration-fired equation, is going to ignore the active channels I was just discussing, the channels whose behavior depends on the voltage. So obviously the flow of ions through a passive channel is still going to depend on the voltage, because the bigger the voltage difference, the more ions will pass through it, but the conductivity won't. The actual channel itself doesn't change its shape or configuration depending on voltage. In the active channels, they do, and those active channels we're not going to, are not part of the Leaky integration-fire equation. So the Leaky integration-fire equation, the equation for the dynamics of the voltage of a neuron that we're going to talk about today, it is an approximation. Well, all equations are an approximation, but this equation is quite a considerable approximation made in that it leaves off these the active dynamics of these voltage-dependent channels. So we're going to deal with the passive channels, and the passive channels allow potassium ions predominantly. Most passive channels of these passive channels are potassium channels. So this is going to allow potassium ions to leak out of the cell. And so the picture we have, or indeed leak into the cell, the picture that we have here is that we have a cell. We're going to discuss, for simplicity, the experimental condition, which is where we have an electrode stuck into the cell, and we can pass current, charge into the cell through the electrode, and charge can pass out of the cell or indeed into the cell through the leak. Just like in the case of the bucket, we had water flowing into the bucket from a tap. That's like the electrode, and because there was a hole in the bucket, water could also flow out of the bucket. And so in this analogy, in the bucket we were talking about water. Here we're talking about the amount of charge. Charge goes in through the electrode, charge leaks out through these passive channels. So that's the charge, but what about voltage? What's the relationship between charge and voltage? Well, voltage we should think of as being a bit like the pressure. If you remember in the case of the bucket, the higher the water, the bigger the pressure, and the pressure determined how fast the water leaked out through the hole in the bottom. Voltage is the same. It's a measure of the force on a charge. In this simple circuit here, you see we have a voltage, which corresponds to a battery, those short and long lines with the minus and plus. That's supposed to represent a battery. I is the current that's flowing around that circuit, and R is the resistor. It's hard for the current to get through the resistor, but the point is the bigger the voltage, the more current flows through the resistor. Just like the higher the water, in the case of the bucket, the more water flows out of the leak. And so the voltage is going to be like the height of the water, or correspondingly the pressure in the case of the bucket. In fact, just like in the case of the bucket, it was important for our equation that the relationship was linear. So the size of the leak was linearly proportional to the height of the water. There's a similar linear law in the case of voltage and current. So current is the movement of charge, and we have Ohm's law, which is that the voltage is equal to I, the movement of charge, multiplied by the resistance, which is some property of the circuit. So in the case of this wire, that would correspond to basically how easily current passed through the wire. In the case of the neuron, it would be how easily the potassium ions pass through the membrane. So it's a relationship between the force pushing them through the membrane and how many, the rate at which they go through the membrane. Okay, so just like in the case of the leak, we had a relationship between the flow of water and the pressure. Here we have a relationship between the flow of current and the voltage. Again, it's given the name Ohm's law. You can see that it's written kind of the other way around to how we wrote it before. So before, with the leak, we had the leak was equal to some number g times the height. Here, and the height is like is proportional to the pressure, and the pressure is the thing that's like the voltage. Here we have it written the other way, where the voltage is equal to something by the current. So for these equations, unlike electronics, it's actually useful to write it the other way around. In other words, we'll divide across by R, we'll let G equals to 1 over R, and we get that the current is equal to G times V. So in electronics, we measure the relationship between voltage and current using the constant proportionality resistance. Here we use 1 over resistance, which we call conductivity. So R is measured in ohms and G in Siemens, S-I-E-M-A-N-S. Okay, so we have, as before, because of Ohm's law, a relationship between the current and the voltage. I is proportional to the voltage, or it's equal to G times the voltage. And so again, this is just the circuit here. The current going around the circuit is given by some number G, which is a property in the case of the little circuit of the wire, in the case of the neuron of the membrane, times the voltage, which is the force pushing the current around. This linear law starts at 0. So if the voltage is 0, the current is 0. That kind of makes sense. If you didn't have a battery, you wouldn't expect any current to go around the circuit. In the case of neurons, that's actually there's a complication. That's not the whole story. And the complication is to do with chemical gradients. Chemical gradients they're a measure of the difference in the number of the concentration of some ion in one place and another. So in the case of the neuron, we have more potassium ions inside the cell than outside the cell. Now ions, they move around randomly because of Brownian motion, because of their thermal energy. They're flying around, they bash into each other, and that causes them to spread out. So it's a bit like children. If you had some children and you drew in chalk a circle on the ground and you put the children inside that circle, very quickly, because the children will run around, bash off each other, ricochet, fly this way and that way, the children will just be dispersed all over the field, and they won't stay inside that initial circuit. As with children, so it is with potassiums. If you had some potassiums inside a neuron, and there was some leaks in the membrane, as there is, then without there being any force across the membrane, even if the voltage inside was the same as the voltage outside, the potassium would travel outwards just because of their random motion, their thermal, their Brownian motion. In the case of the children analogy, we could supply a counteracting force. So children are attracted to dogs, much as they hate pigeons. These are one of the defining properties of children. And so if I put a dog, in this case Snoopy, in the middle of the circle, the children will be attracted to the dog and will end up back in the sort of original area. And they'll continue to move around, but their diffusivity, the effect we saw before where they just sort of spread out over the field, is balanced against against their attraction to the dog. And so what we're seeing in this silly analogy is that the chemical gradient is a measure of something that's like a force. So there's a diffusion, and to counteract the diffusion, you need a force. Or you can counteract the diffusion with a force. So the chemical gradient causes something that looks a bit like a force. It's an inclination towards movement because of random motion and the fact that there's more children, more something in one place than another. So again, here the children, or the potassium ions are in the middle. There's no force on them, but just because they're moving around and they're in the middle and there's none outside of the circle, on average, because of that moving, it will cause them to become more spread out as time passes. That's something that you can balance with a force, and if you have a force, then they can remain not spread out. So it is with the neuron. So the neuron has potassiums inside and not outside. And so if there was no force across the membrane, there would be a flow of potassiums out of the cell. And a reciprocal if the flow of sodiums into the cell modulo the complication that these ion channels are ion selective. And so because of the gradient, if there was no force, as I said, we'd see this flow of potassiums through the cell. And so to counteract that, in other words, we saw in Ohm's law, in the Ohm's law that applies to batteries and circuits and electrons, that when there's no voltage, there's no current. But in the case of in the case of ions in a neuron, you need there to be a voltage difference to counteract the diffusive pressure. In other words, the place where there's no current is not when the voltage is equal to 0, but when the voltage is equal to some other value, which you can calculate based on your knowledge of thermodynamics and the laws of diffusion, etc., but which we're just going to call EL. Okay, so the leak potential, unlike in the case of the bucket, doesn't have a zero leak for a zero value of the quantity we're interested in here, the voltage. In fact, the law looks something like this. The leak current is equal to G, not G times V, but G times the difference between V and EL. And this EL, as I said, it's in principle calculatable if you knew the difference in the concentrations of the ions inside and outside the cell. This EL is known as the leak reversal potential. You'll notice I've gone to a minus V here. It's very easy to get confused about the signs when you're thinking about this story. What the expression was for, in this case, was for a current inwards rather than outwards. But what I'm trying to say is don't worry too much about the signs. I'll make sure it's right. And if you start to think about it a little bit, you'll get confused. If you think about it a lot, you'll see that I'm correct. So try not to worry about the signs. So in summary, this is the leak conductance. I'm going to call it I sub L just because it's the leak conductance. It's the thing that corresponds to the leak in the bucket equation. It's the leaking into or out of the cell of charge. In this case, charge carried predominantly as the charge on a potassium ion, but that doesn't matter. The main thing is it's the leak of charge in and out of the cell, and it's given by some constant, by EL minus V, where EL is the leak reversal potential. And rather than using a big G for this specific conductance, we sometimes use the symbol, or often use the symbol G sub L, where again the L is standing for leak. Secondly, if you remember in the case of the bucket, there was a relationship between the height and the volume of water. Here, the amount of water corresponds to the amount of charge, and the height corresponds to the voltage, and we'd expect there to be a relationship between them. In fact, there is. It's called Farad's law, or the law of capacitance, and it says that the amount of charge, is proportional to the amount of voltage, and that constant proportionality is called the capacitance. And so that's the sort of second component in our equation. It's a relationship between charge, because current is a rate of change of charge. Current carries charge, just like the inflow of water carried water, and voltage, the thing that upon which the size of the leak depends. Okay, so now we can write down the equation. On the left-hand side, we have the rate of change of charge. How does charge change with time? And then the right-hand side, we have the things that change the charge, the current coming in and the leak going out. And we have expressions for all of those things. So the charge is Q, and the rate of change of charge, how does charge change with time, is dq dt, and that's equal to the current coming in the case of the experimental setup through the electrode, in the case of neurons, in the brain, predominantly from the dendrites. The charge ultimately comes from interaction at the synapses. Either way, we have some current coming in, but we also have some current going out, which is this I sub L. We're going to call the electrode current big I sub E. That's just traditionally what it's called. The E is standing for electrode, obviously. And since Q is equal to CV, and C is a constant, as we saw in the case of the bucket equation, we get C dV dt. We know what the leak looks like. It's GL EL minus V, asking you to trust me on the signs. And then coming in, we have the electrode current. As we did with the bucket equation, it's nice to divide across by the G, in this case the GL. So we call tau m is equal to C over GL. It's called the membrane time constant. You will remember that tau looks like a time constant. And so tau m is the time constant associated with the neuron. It's one of the basic properties of neuron. People who are studying neuron, a particular neuron type, one of the first things they'll do once they start doing electrophysiology is to measure the membrane time constant. And then when we divide across by the G, if you remember before, we ended up with 1 over G's everywhere. It's traditional here to write Rm for 1 over GL. So Rm is the membrane resistance. And again, it's the membrane resistance. It's one of the passive properties of the cell, and it's one of the things that people measure when they are studying a neuron type. So that's our equation, tau m dV dt is equal to EL minus V plus RMIE. And that's the Leaky integrator part of the integrating fire equation. It is, of course, the same, almost the same equation that we had before. There's just this extra constant, EL, coming into it. So before we had I over G, now we have EL plus RM by I. But you can see that that's, as far as solving the equation is concerned, studying its dynamics, that's no big change. So we've more or less seen this equation before. And we can solve it if the current is constant. So for constant input current, we'll have V of t is equal to EL plus RMIE, that's the constant on the right hand side, plus V of 0, some value that we're starting off with, minus the same thing, EL minus RMIE, multiplied by e to the minus t over tau m. So just like in the case of the bucket equation, V tends to go towards some equilibrium value, EL plus RMIE, and it goes towards it by exponential relaxation, according to e to the minus t over tau m. Now, as I said before, as anyone will tell you, that isn't the full story. So in the membrane, there are these passive channels, and we've modeled them with our EL minus V. But there are also the active channels, the channels that are gated by voltage, whose conductivity changes according to voltage. These are complicated, amazing molecular machines. They're part of the great wonder of nature that these things could evolve and facilitate the behavior of neurons that we require as a substrate for thought and action. A picture of a potassium channel is given there from Wikipedia. The main point is that these channels change their conductivity in response to voltage. And what they actually do, among the things they do, is when the voltage gets close to some threshold value, sodium channels start opening. That allows sodium to flow into the cell because of the chemical gradient. That will push the voltage up further. Those sodium cells will close, but potassium channels will open. That will stop the influx of sodium but permit the outflux of potassium, which will push the voltage back down. In other words, what we're leaving out in this this leaky integrator is an event or a behavior where, as the voltage goes up, suddenly the changes in voltage aren't being driven by the input current, but rather by new currents that arise out of the opening and closing of voltage-gated channels. And the effect of that is to cause a spike. This is, of course, a spike recorded by Hodgkin and Huxley when they were first trying to understand the dynamics of neurons and wrote down their famous equation for the dynamics of neurons. What you can see there is the voltage of the neuron suddenly shoots upwards and goes downwards and forms a spike. And these spikes then propagate along the axon and allow the neurons to communicate one to another. But these spikes depend on stuff that isn't in the leaky integrator. They're depending on conductances that we haven't included. The only conductances we've included are the ones that are the passive conductances that give us the leak, the EL minus V. And so to include the behavior that creates the spike, we would need the things in red there, the currents from the gated channels. And so in the leaky integrating fire equation, we kind of put those in by hand. We pretend they're there, but we don't include them directly in the model. In other words, we add an extra line to the model. We say, this is how V behaves. tau m by dv dt, the rate of change of v, is equal to EL minus v plus RMIE, with the extra instruction that if v ever goes over a threshold value, Vt, then there's a spike and we reset the voltage. So if you look back at this behavior here, after the spike, the voltage is pushed back down to a reset value, which is typically quite close to EL, the leak value. The leak value, I should have said, is around minus 70 millivolts as well. So when the voltage goes over a threshold, we say, well, that's the place where this equation doesn't work. We're not going to model that. We'll just say now a spike has happened. If we were simulating this neuron as part of a network, we would then in our code have to have some line of code that says this neuron is spiked, tell all the other neurons that this neuron is spiked, and if they're connected by a synapse, including now in your modeling the change in currents at the synapses, that would be the consequence of that neuron spiking. If we were trying to run this model just to model some particular neuron and we were trying to match its spike train, then we'd say, well, these are where the spikes are that are predicted by the Leaky integrator and fired equation. So in other words, we mark down in our simulation that there's been a spike at this point, but as far as the actual model is concerned, what we do is we set the voltage back to the reset value. Okay, and so that just says again what I said. Vt is called the threshold, Vr is the reset value. And here I'm just giving some typical example of values. So the reset value, as I said, is minus 70 millivolts. In simple models, we often take the reversal potential EL to be also minus 70 millivolts. In more sophisticated models, you might have the reset for a particular neuron type. You might have the reset value higher or lower. And then the threshold value is minus 55 millivolts. Again, that's just a typical value. And if you were trying to model a particular neuron type, you would try and use values that were special to that type of neuron. Okay, so let's look at the behavior of this model neuron a small bit. I'm going to write V bar for the equilibrium value EL plus RMIE. I'm imagining that we have a constant current, so I'm looking at how the neuron model behaves. If we were imagining doing an experiment where we've introduced an electrode, we're pumping charge into the neuron at a constant rate, and we're looking what the neuron does. This is the solution. V of t is we get is V of t is equal to V bar, equilibrium value, plus V0 minus V bar e to the minus t over tau m. So as we discussed before, the V0 minus V bar is multiplying something that's going shrinking away to 0, and so Vt is going to go to V bar. And so if V bar is less than Vt, and we imagine V0 might be, that might be the value just after spike, so that might be VR or something. Then as time passes, the neuron's voltage will head up towards the threshold value, up towards V bar, the equilibrium value. It'll never reach the threshold value and it'll never spike. It'll do something like this. So if you look in the GitHub where these nodes are, you can see a little Python script that runs this model. So using the methods I described before, we simulate this neuron using Euler integration. If you look, you can see what the constants are. We have tau m is equal to 10 milliseconds, for example, and RMIE is equal to 12 millivolts. IE is a current, RM is resistance. If you multiply the two together, you get effectively a voltage. And with that value, V bar is less than minus 55 millivolts. And so we can see here that the voltage is heading towards its equilibrium value, but it's never crossing it, and so the neuron never spikes. Conversely, if V bar is greater than Vt, then as the V heads towards its equilibrium value, it's going to reach the threshold value, and then there's a spike, and it will get reset. And so if for currents, for values of IE that make EL plus RMIE greater than the threshold value, in this case minus 55 millivolts, then the neuron will keep trying to reach the threshold value. Every time it approaches it, it's going to get reset back to the reset value. So the equation, the differential equation bit, only applies between spikes, because at the spikes, V is reset by hand. And so we'd start the equation again with a new V0. Okay, so what you see happening here. So here, just to put this spike, I put the spike in by hand by at one time point, setting the voltage equal to 20 millivolts, which is a sort of a range a spike can reach. And here you can see in dotted the trace I talked about before, where the equilibrium value is lower than the threshold, and then the trace where the equilibrium value is higher than the threshold. You see the neuron that's heading up towards its equilibrium value. On the way, it reaches the threshold, there's a spike, it gets reset. And so you can see the neuron goes from not spiking, and then as you increase the voltage a small bit, the input current a small bit, you go to regular spiking. You can actually map out the firing rate against input current curve, the so-called FI curve. Again, always an interesting property of a neuron that you might measure using electrophysiology if you're looking at the behavior of a new type of neuron. And you can see that in this case, until RMIE reaches 15 millivolts, there's no spiking, and then once it reaches the threshold, this special value 15 millivolts, the firing rate rises quite quickly. And so that's a behavior we see in neurons, this no spiking and then all of a sudden spiking. That behavior is modeled quite well by these integrate and fire neurons. And so what we've learned is that what we've discussed is a simple model of neurons, the leaky integrate and fire neuron. It has some dynamics which are like the dynamics of the bucket, plus this extra bit where we say if the voltage reaches a threshold, there was a spike. That tells us that the behavior of neurons a bit like this. It shows the same sort of behaviors that the bucket showed. It shows this sort of filtering of the input, with the extra caveat that you have this firing behavior as well. So you have the input being filtered, but whenever the input causes the neuron to reach the threshold, you get firing. We also saw that the model shows some of the properties that would be typical of a neuron's FI curve. In other words, there's no firing until you get to some special value, and then it starts firing. It doesn't show other phenomena. So neurons often show spike rate adaptation. In other words, if you keep going with the same constant input and it's causing the neuron to fire, the firing rate will fall off a little bit as time passes. That's not part of the model that we just looked at, but you can add extra bits to the model to capture that behavior. So it's easy to include adaptation in the neuron. The neuron also doesn't show any sort of realistic behavior near threshold. As a real neuron gets towards threshold, before the nonlinearities, because of the voltage-gated channels, really take over and cause a spike, they're already starting to have an effect on the behavior of the voltage. But again, there are additions you can make to this model that start to capture that behavior. In other words, this model is maybe the simplest spiking neuron model. It captures some properties, the spiking of the neuron, the property of the FI curve, where you have nothing until the current reaches a critical value, after which you have regular spiking. So it shows some of those behaviors. It doesn't show many of the behaviors that we see neurons having, but there are elaborations in this model that capture those phenomena. In fact, you can think of the Hodgkin-Huxley model, the full model, which includes the full dynamics of the spiking, as an elaboration of this model. So this is the first in a sequence of more and more complicated models, which ultimately brings you to the Hodgkin-Huxley model. The Hodgkin-Huxley model is a model very built on a similar philosophy of this one. You're trying to look at the currents that go through across the membrane, but you include not just the passive current that we've included, but all of the nonlinear voltage-gated channels that you might find in the neuron. That gives you a much more complicated model, one that's much harder to solve, but can be treated with the same sort of numerical methods we discussed in the last lecture. And that model will capture, in fact, the production of spikes and can quite accurately capture the behavior of neurons. Even that, of course, doesn't. The Hodgkin-Huxley neuron leaves off some complicated phenomena to do with the actual shape of the neuron itself. So, for example, this neuron has calcium channels. The concentration of calcium can vary quite a bit across the neuron, and that can cause position-dependent changes in the voltage, and so you can no longer treat the neuron as having a single voltage inside. But there are, again, elaborations of the Hodgkin-Huxley model that start to include that. If we were modeling networks, this is only part of what we need to do. We need to know some model synapses. In fact, interestingly, many synapse models use very similar equations to the one that we've been dealing with, the equation that we described initially as the equation for a leaky bucket. And it should also be noted that in the full Hodgkin and Huxley model, the dynamics of the voltage-gated channels also in many ways resembles the leaky bucket equation. So this first order differential equation that we've studied and we've used to study the neuron in the Leaky integrated fire model is also part of the mathematical description we'd need to model synapses, or indeed to extend the Leaky integrated fire model to include other channels, the voltage gated channels. Numerical calculations, just as a quick note at the end. Obviously, the more complicated these equations become, the harder they are to solve exactly. We discussed before that you might solve them on a computer numerically. You can do that with MATLAB or with Python or Julia. But there are also specialized packages, Brian in Python or Genesis, Neuron or Nest, that are well optimized to integrating the sort of equations that occur in neuroscience. So to summarize, we looked at the dynamics of a neuron. Because we were leaving off these active channels, we were mostly modeling the dynamics that the neuron shows away from the threshold. We discussed chemical gradients, I think, which is a crucial aspect of how neurons behave. It was crucial to understand why there was an EL, the leak reversal potential in the integrated fire equation. The same sort of discussion is necessary to understand how the the ion selective voltage gated channels that power the spiking behavior of the neuron behave. We looked a little bit about at the integrated fire model. We discussed the addition of the spikes by hand, as it were, and we saw that the integrated fire model can produce an FI curve that's somewhat similar to that seen in real neurons. Thank you.